The primary objective of the method presented in this video article is to rapidly generate spatially defined cell patterns to facilitate in situ spatiotemporal analysis of cell-to-cell interactions.
The presented strategy leverages the microfluidic probe technology. The microfabricated probe head localizes nanoliter volumes of biochemicals on biological substrates to pattern cell monolayers. The main advantage of an MFP-based patterning technique is that it is nearly instantaneous owing to local lysis, allowing real-time modification of the patterns to be generated.
We first had the idea for this method when we noticed that other methods to generate cell patterns require long, multi-step protocols before actually seeing the success of the cell patterns.
To run stable experiments with a hydrodynamic flow confinement on a cell surface, the MFP parameters need to be optimized. For this purpose, visual demonstration is critical.
Aditya, Kashyap and Julien course will be demonstrating this procedure.
The operational modules of the platform comprise motorized syringes, motorized stages, and a controller. The MFP is connected to a motorized Z-stage to control the gap distance between the head and the substrate, and the substrate holder is attached to the X and Y motorized stages constituting the scanning system. The MFP head has fluidic vias to connect the pumping station, mounting holes to mount the head onto the Z-stage, and channels that exit the polished apex. The apex is set coplanar to the cell culture substrate. To prepare the pumping station and fluid handling apparatus, clean syringes